Okay, Tanvi, are you feeling comfortable right now
Yeah, sir.
and ready for your mock interview?
Yeah, yeah.
Okay, so just introduce yourself in a brief so we can start further.
Yes, sir. Good evening, sir. My name is Muhammad Tanvir Anwar, and I am a native of Bihar. And I have done my schooling from Mount Sinai District, Kerala. After completing my schooling, I moved to Kanpur in Uttar Pradesh, and I have completed my bachelor's in biotechnology from Chhatrapati Savaji Maharaj University, Kanpur. And after that, I moved to Lucknow, and I have completed my master's in biotechnology from BBAU Central University in 2023. After that, I have been working in a DST SOP project from the past 5 to 6 months. From November 2nd, I have joined there. Then my hobbies are having to play sports in the evening because I was a sports enthusiast also. Apart from that, talking about the academic achievements, I have qualified DBT JRF examination, and I have also qualified BARC examination for a stipendiary training, and I appeared for the final interview. And I'm right now I'm waiting for the result. And patent examination also I qualified, and I'm waiting for the final interview.
Okay, well done, Tanvir. So basically, you are in a project, right? Right now.
Yes, so I'm working on a project. As I was not able to qualify the category one, so now I'm working on a project.
Okay, got it. Means you have a category two DVT certificate, right?
Yes, sir.
That is
Yeah, yeah, yes.
okay. Okay. By on behalf of that, you can only just join the project under the DBT funded colleges or universities or such
Yes, sir.
centers.
And if the PI, yes, sir. And if the PI is agreeing, then it can be converted to a PhD.
Okay, so what is the title of your project? Currently, you are doing that.
So actually, project title is too long, but it is basically on Helicobacter pylori mediated infection, and then we are using some probiotics to study the immune modulation. Exact top heading I cannot recall now, but it is basically on the H. pylori infection in THP1 cells, and we are studying the immune modulation. So now the lab is not set up as the professor is very new. So now I am currently working on some in silico work on meta analysis.
Okay. Tanvir, as you have
Yes.
filled your application form and mentioning there that you are interested in the RDT, means recombinant and technology, immunology, and genetic
Yeah,
engineering,
sir.
right?
Yes, sir.
So, can you suggest some name of a GMO food,
The
genetically
GMO
modified?
food, yeah, sir. Now currently there was a journal, not food we can say, but as a substitute we can use like mucorized CTP. You might have heard. So mucorized CTP is a cholera vaccine. So that is produced in the rice. So that is also a genetically modified food, and it can also be act as a substitute for the vaccine. So edible vaccine concept was there. So this one is the very latest research of 2021. Apart from that, golden rice is the very common and famous thing which we are hearing from many years back. So these two things I can recall right now.
Okay, have you heard about the eggplant?
Eggplant. No, sir. Sorry. Eggplant.
Okay, 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 no issue, Tanvir. We move ahead. Have
Yes,
you
sir.
heard about the prebiotics and probiotics? Nowadays, these terms are trend, and
Yes, sir.
hopefully, yeah. So, what is this?
Yeah,
That
sir.
is, yeah.
Pro Probiotics are basically some microorganism which are residing in our body and acting as a friend to our body. We can say in a very simple words, like they are acting as a agent which are helping our body to maintain the homeostasis of our particular that gut area.
Okay, and what is the prebiotics?
Prebiotics. This term I have heard, but right now I'm not able to demarcate.
Okay, leave it. Nowadays, so many microbiologists are focusing on the gut microbiota, What do we
Yes,
understand
sir.
by gut microbiota and how it is beneficial for human well health?
So, as gut microbiota and as my project is also on this, so basically in our gut area, different microbes are residing in our body. So the major research gap which we can see is that in this area many research has been done, but a new concept has came that how we can use the probiotics or the gut microbiota modulation as substitute for antibiotics. One concept is there if in my project. So basically what we are searching and what the current trend is that we are checking that how these microbiota which are living in our gastrointestinal area or the gut area, how they are having certain modulation on our immune system or certain signaling they are modulating or any kind of interaction which is happening in that area. So once we are able to decipher the things that which is signaling or what is happening in that particular area, we can have a substitute or we can get certain results which may be beneficial in the coming years. Like suppose my project on which I am working, suppose if we are getting we are testing certain probiotics or probiotic bacteria, that is actually a secret as part of the project tool. But once if we are confirming that particular probiotic or particular microorganism is modulating some immune response and leading to certain beneficial action in our body, so it can be published and it can be patented also that these are the things which can be used as a substitute for antibiotic resistance or certain microbes can be helpful for our body. So these kind of things can be deciphered in the coming years.
Okay, Tanvir, obesity is one of the major
Yes,
issues in so many countries, and so many people are suffering from obesity. Is it possible
yes, sir.
to cure the obesity or to rectify the problem of obesity by using gut microbiota?
Sir, as what I can recall, I don't have much idea, but somewhere it is related. Even I am not able to explain it properly, but somewhere in some journals I have read some articles. So I think it is possible if we are modulating and if we are checking the different interaction which is happening in the gut area. So obesity may be controlled.
Okay, that
Yes,
means
sir.
right now you don't have enough data about this, so
Yes, sir. Yeah.
no issue. We will move on further. Okay, Tanvir, in microbiology, for the
Yes, sir.
maintenance of the culture, that is microbial culture or fungal cultures, there are certain way and there are
Yes.
certain rule. Can you suggest some
Yeah.
technique or some way to preserve these microbes, that is bacteria, for a long time, like as year by year?
Okay, so actually, I'm not very much fascinated towards microbiology, but still, certain techniques I have seen as I have joined the project. So, one slant we can prepare and we can store certain microbes. Apart from that, liquid nitrogen minus 196, we are keeping our stocks. Then minus 80, we can preserve our stocks or stock samples. Then and certain roller tube techniques are there for the anaerobic bacteria, which are microbes. For that roller tube techniques are there. Then these few things I can remember now. So,
Okay, well done. Tanvir, probably you have heard about the fibers. Fibers are very good for nutrition and very good for the health, right? So, how these
mhm.
fibers help or improve the digestive tract or digestive system?
Digestive system.
Yeah, probably you have heard about the talking about the fiber containing food suggestions for the
Yeah, yeah.
body
Yes,
health,
sir.
right? So, how
Yes,
fiber
sir.
containing food will help the digestive system?
So this thing right now I'm not able to
Okay,
explain,
is it
but
possible to
definitely I will
digest the
look into it.
fiber?
The fibers usually like as we have some data which I can recall. Certain fibers we cannot digest that ruminants can digest, like a cow can digest. So the excess amount of fibers if you are consuming or some fibers which are not digestible by your body will not be digested. But certain fibers are still recommended as in food. So exact thing I'm not remembering why they are recommended.
Okay, in recombinant DNA technology, there are certain there are many enzymes used in preparation of any clone, that is either for the prokaryotic or
Yes,
eukaryotic,
sir.
right? So,
Yeah, sir.
can you
Yes,
name
sir.
that enzymes which are involved in the cloning purpose?
Yeah, sir. So actually, when we are doing the cloning, the first enzyme which come in our mind is restriction endonucleases. So for that, restriction endonucleases are very much beneficial because we it is acting as a molecular scissors, and we are cutting the different sections of our suppose DNA. If we are working for different sections of our DNA, we are cutting based on the restriction sites, that is 5 to 7 nucleotide base pairs. So that is a particular restriction endonuclease is recognizing that particular sequence, and on that sequence it is cutting. So it is acting as a molecular scissors. So restriction endonuclease is one thing. Then DNA polymerase, obviously we need, and to be very specifically, Taq polymerase or PFU polymerase when we are working in the in vitro condition. Then phosphatases, alkaline phosphatases, then TDT transferases. These are the enzymes. Then ligases. These are the major enzymes which we are using in the recombinant DNA technology when we are preparing some recombinant molecule or any cloning we are doing.
Phosphate.
And suppose for on mRNA, if you're working and you want to prepare cDNA, then in hello.
Okay, uh, Tanvir, phosphatase
Hello.
and kinase. These are the two enzymes used in regard with the phosphate. What is the function of a phosphatase and kinase?
So phosphatase will actually remove the phosphate, and kinase will add the phosphate.
Okay,
So the basic logic behind this is like when we are working in a lab, so we are adding phosphatase because when once the phosphate group and 3-OH group is there, so that may religate and our recombination will not take place. So in order to avoid that, we are treating our sample twice so that the phosphate group will be removed. So both side will be OH, so it will not be ligated.
okay. Tanvir, suppose you are dealing with a fragment of a DNA to be cloned, and you transfer that DNA fragment in a plasmid vector, and that recombinant plasmid will transfer in a host cells for the multiplications, and you want to screen out that the that is targeted DNA will transfer or not. So, what will be the possible technique to use or to screen out the recombinant molecule or recombinant plasmid? how how you will
Sorry, sir,
confirm
I
that?
was
Yeah.
not able to hear you properly. Hello.
Okay, my question was that.
Sir, I was not able to hear you properly. Can you
Okay,
please
okay, got
repeat?
it, got it. I
Yes, sir.
repeat my questions right now. I'm audible to
Yes.
you.
Yes,
Okay.
sir. Yeah.
Yeah, I was asking that. Uh, Suppose you are dealing with a fragment of a DNA to be cloned in a plasmid vector, and you transfer
Yes, sir.
that segment of a DNA in a plasmid, and after that, that recombinant plasmid transfer in a host
Yes.
cell for their multiplications. To check out or to screen out that
Yes, sir.
transferred plasmid DNA, there are certain method. So, can you name that method which are used for the screening of a recombinant plasmid
Yes,
or
sir.
recombinant vector?
So, yes, sir, definitely. Actually, when we are doing any screening for cloning purposes or any recombinant in any recombinant DNA technology, so basically we are doing the two-step screening. First one is transformation screening, and second one is recombinant screening. So, and in the plasmid or whatever the clone we are preparing, so not clone, construct which we are preparing. In that we are using at least two markers. So one will be for screening, and sorry, one will be for transformation, and second one will be recombinant. For example, like LAC-Z, that beta galactosidase activity is one of the major recombinant screening technique, that blue white screening we are calling it as. Like when we suppose we are inserting our gene in between the LAC-Z gene. So once the LAC-Z is getting inactivated, it will not react with X-gal, which is the substrate, and once it is not reacting, it will not give a blue precipitate. So that blue white screening concept is there. So based on that color we can say that our particular gene is transformed to that particular location or not. Apart from that, for the normal transformation screening, we can use some antibiotic like ampicillin or tetracycline. So this kind of ampicillin or tetracycline, any antibiotic resistant gene, if you are using, so we can do the transformation checking by applying that particular antibiotic. If the particular clone is not resistant to antibiotics, so we can say that that particular gene is being transformed, because the activity of that particular antibiotic resistant gene will be lost if our gene is incorporated in that particular area. So
Okay, well
like
done.
this we can
Well,
do.
well done, Tanvir. Have you heard about the inactivation method of screening for the recombinant or transformed plasmid? Inactivation method.
Yeah, gene active inactivation method. Yeah,
Yeah.
so that's what I explained previously. In suppose chloramphenicol antibiotic resistance is there, so in that particular gene, if we are inactive transforming our gene, so that particular gene activity will be lost. So that phenomenon is called insertional inactivation, because we are intentionally abrupting the particular gene function. So if the gene is not working, we can say that our gene is transformed to that particular.
Good. Tanvir, antibody is one of the key
Yes,
molecules which play a very important role in the immunology, right? And it is a functional
yes, sir.
protein. So, what is the different level of structures involved in formation of a functional antibody?
Different level of structure.
Yeah, different level of structures involved in formation of a functional antibody.
The actually different level of structure. What you mean to ask? I'm not getting that, but I
Like
will try to
yeah,
explain.
like there are four different level
Yeah.
of the structure of the protein, like the primary, secondary, tertiary, quaternary, etc.
Ye-
So in
Yes.
respect of that,
Yes.
what different kinds of a structural level are there involved in the formation of a
Yeah.
complete functional antibody?
Yeah. It will be in the tertiary structure. So as far as I know, because when we are talking about antibody, it is having the four chains. We can say two light chain and two heavy chain, along with 16 sulfide bonds are there. Sulfur bonds are there. 16 sulfur bonds are there. And in the heavy chain and light chain also, we can demarcate like constant region and variable region. So in the constant region, the particular constant region, when we are talking variable region is differentiating, or it will be changing. Variable region, we are saying the upper part of the antibody. And in that region only, the antigen will come and interact and leads to the further immune responses. So as far as I know, it is in the tertiary structure.
Okay, Tanvir, have you heard about the Genome India project?
Genome India project. Yes, so somewhere I have read.
Yeah, what is this?
Recently, DBT has launched one website also for this, but exactly very fluently I cannot explain, because I'm not getting the exact idea. But somewhere I have heard DBT India has launched one website, and in that they have uploaded all the data which they have sequenced.
Okay, no
Yes,
issue, Tanvir. It's obvious for everyone that all the things can recall in mind at the time of an interview. So, no issue. We will move forward. Uh, for the formation of antibody, there are certain conditions
yes.
required in the body. Suppose all the times our body do not produces antibody. So, what is the
Mhm.
conditions and what is the situations to form and to produces antibody for our body? In which conditions our
So
body make
basically,
antibody? Yeah.
yeah. So basically, whenever there is an antigenic interruption, or whenever an antigen entering in our body, in that time that antigen will majorly interact with the B cells receptors. So when the B cell receptor is interacting with that particular antigen, so based on the different type of antigen, suppose if it is a proteinaceous antigen which is T dependent or T independent. So if it is a T dependent, for example, we can talk first this. So if it is a T dependent antigen, so it will come to that particular B cell receptor, then B cell receptor will internalize that particular antigen, and after processing it to some 10 to 15 base pair, it will present through the MHC to the T cell receptors. And when the T cell receptor, MHC, and the antigen is interacting all together, means B cell and T cell is interacting together. So T cell will provide some co-stimulatory signal through B7 and CD8 if it is a viral antigen, or CD4. So this co-stimulatory reaction will happen, and this T cell co-stimulation will lead to the differentiation of B cell as memory cell and plasma cells, and this plasma cell will be further signaled through certain cytokines to produce the antibody. So basically, the plasma cell will produce the antibody, and if it is a T independent antigen, still T cell will direct that particular B cell for the antibody production, but the plasma cell or memory cell will not be.
Okay, good. Well done, Tanvir. As we know, antibody has ability to neutralize the impact of antigens, right? Okay.
Yes, sir.
So
Yeah, yeah.
my question is that how any antibody can neutralize the impact of an antigen? What are the mechanism behind it that is an antibody can neutralize the effect of antigens?
So one example which I can recall now, we have heard about ADCC, antibody-dependent cell-mediated cytotoxicity.
Very good.
So whenever a antigen is entering in our body, so that antigen will interact with the antibody, and in the antibody, the FC region will interact with the receptor present on NK cells, and this NK cell will lead to the killing of that particular antigen. So this can be one example of neutralizing the antigen which is entering in our body through ADCC phenomena.
Okay, what is the key functions of antibody?
The key functions of antibody, it majorly it is interacting with the antigen. This is the basic idea which we all know. Apart from that, antibody have certain different role in the immunity itself. Like IgG, if you are talking about, it will crosses the placenta and it will provide immunity to the new net. Then one thing, as I told, the ADCC is there. Then antibody will also uh, what we say, antibody will also provide certain different kind of immunity, we can say, basically, like antibody will interact in different area based on the location. The antibody type is also changing. Suppose it is in circulation, so IgA will be there. If it is the first immune response is happening, IgM will be there. So based on different location and different condition in our body, different kinds of antibodies are there. So majorly the role of antibody is to recognize the antigen, and the further killing process or further neutralizing process is carried out by some other phenomenon. But major role which we can say that antibody is basically responsible for identifying, or it is acting as a police to identify whatever barrier has been crossed by the antigen and it is entering in our bodies.
Good. Is there any role of antibody in hypersensitivity reactions, Tanvir?
Yes, sir. Hypersensitivity reaction IgE is involved. So whenever the first interaction is happening with any allergen or any hypersensitive molecule, so the mast cell will get mast cell that will get sensitized, and in the second infection or second attack of the antigen, that will degranulate, and certain antihistamine or certain effector molecules will be produced, which leads to hypersensitivity reaction.
Good. Immune tolerance is a term. My question is that
Yes, sir.
immune tolerance is good or
Yeah.
bad for us? That is beneficial or harmful?
So, if we are talking from the point of clinical view, it depends on what topic we need to talk. Suppose immune tolerance is there, so the immune tolerance is basically that the particular immune system of our body will not harm our own cells. It will recognize our own cell and it will not respond to it. So, suppose transplantation, if you are talking, so in that or any organ is getting transplanted in our body, so immune tolerance, if as we talk, so immune system will recognize it as a foreign. So transplantation failure will occur. But on the other part, if we are coming up with some ideas in which we are fooling our immune system. Like many transplantations are occurring and many immunosuppressants we are eating. But if we are able to find a substitute, such as like immune privilege sites are there, and that sites certain antibody will not be recognized, that and immune response will not be generated, cornea and uterus, this kind of area. So it depends. If the body is recognizing a external agent in our body, it will harm it definitely. But the clinical point of view, if we are talking, immune tolerance will be negative for the doctors, because when they are doing a transplantation, it will lead to the rejection of the graft. But if they are coming with certain substitute, so they can go for it. But talking about the normal phenomena, when some antigen is entering in our body, so our body will recognize that as foreign, but our own cells it will not recognize, so it will not harm our own cell. So autoimmune diseases which we are talking about, so that will not happen.
Okay, well done. How many different kinds of antibodies are there in our body?
So there is one mnemonics I can recall now. Gamed we are saying IgG, A, M, D, and E. So they are based on the order. As I have told, the highest titer is for G, then A, M, D, and E. So 80 to 85% is G, then 10 to 15% is A, then M is 5 to 10% is, then D is 0 to 5, then E is the least. So D is the least one, 0.2 to 0.5. That range it is like.
Complement system is an integrated part of our immune system. This is also involved in the immunity.
Yes, sir.
Antibody
Yes, sir.
has ability to activate these complement systems. If I ask to you,
Ye-
among these five different antibody, that is antibody G,
Yes, sir.
A, M, R, E, and D, etc., which one
Yes, sir.
has good and which one has high affinity to activate the complement system? So what will be the answer?
So IgG will be the answer if you are talking in the umbrella sense. But to be very specific, whatever I can recall, IgG3. These are the G3 is the best complement activating antibody. But when we are talking normally, IgG is the most potent activator of complement system, we can say.
And what about IgM?
IgM can also activate, but IgG is the most potent, we can say. That is having the high affinity for activating the complement system.
Okay, what is adjuvant?
So adjuvants are the, we can say, any agent which is enhancing the immune response. Like suppose one vaccine we are taking. In case of vaccines, we majorly we are using adjuvants. So to enhance that particular immune response, we are using adjuvant. It is not acting as an agent to enhance to establish an immune response, but it will help the particular vaccine or particular antigenic material which we are taking in our body as a vaccine to enhance its response. Like it will enhance the timing of diffusion of that particular antigen in our body. Then post immunitary signals will be enhanced. So adjuvant is actually helping as a plus point for the vaccines when we are talking.
Okay, well done, Tanvir. Tanvir, you are from Bihar, right? So tell me some
Yeah, so
beautiful things about the Bihar. Why the peoples will go there? So can
yeah.
you? Yeah.
So I born in Bihar, but my whole life I have been spending in Kerala, as I told earlier. But in Bihar, as we have studied the history in our early classes, so it is a we can say center of the Indian history. We can say so the modern Indian history, which we are reading the whole independence struggle and the advent of Gandhiji in India. So all the from the monumental point of view, from the historical background, if you are looking, Magadha, Patliputra, we have studied. Then certain big dynasties were there. So somewhere Bihar is having that particular glory, we can say. But now we can say that Bihar is not much developed, but still there is too many historical memories which is associated with Bihar. And now when we are going to Bihar as a tourist, so many historical places we can find. Some of which I can recall is Vikramshila University. Then Rajgir, some glass walk is made recently. So these two three things I can remember. And as I am from Ara, so Veer Kumar Singh we have studied in history, so his quotes are there in Ara, so this different
So
things are there.
currently you are in a Kerala, right?
No, so actually now I am in Lucknow. I am in the same university from where I have done my masters. I am doing the project in the same university.
So okay, currently you are in Lucknow. So tell me some
Yeah, sir.
beautiful things about the Lucknow.
Lucknow, sir, as we are calling it as Nawabo ka Sahar
Yeah.
in Hindi, we are calling it as. So one thing which I have felt from the last 2 3 years is the people of Lucknow are quite good. They are very friendly. They are respecting others, and they are very kind-hearted. This thing which I have observed in my 2 3 years, I can say. Then certain foods are very good in Lucknow.
Okay. So okay, good, Tanvir. So it's I think it's over now. So right now it is your turns to whatever you want to ask regarding your interview, regarding your preparations, and whatever you have of questions. So now it's open for you. So you may proceed. Yeah.
Uh, So can
Now
I
you
switch
feel,
to
now you
Hindi
can
now
feel
or
comfortable, and
yes?
you can talk in Hindi as only because your interview session has been
Yeah, yeah.
complete now. Yeah. Feel comfortable.
So actually, sir, yeah, actually, the one problem which I am facing is like I am not stopping for is or listening the interviewer. जो interviewer बोल रहे हैं, कभी कभी ऐसा होता है कि बीच में पहले मुंह से निकलने लगता है, ये मेरे को फील हुआ है कभी कभी. एंड सेकंड थिंग इज आई एम नॉट ए वेरी इंटरव्यू बहुत बार मैंने कभी नहीं दिया है, तो थोड़ा सा एक फियर रहता है कि इंटरव्यू में पता नहीं क्या बोलेंगे. और कभी कोई क्वेश्चन करता है तो एकदम से इजी क्वेश्चन ब्लैंक पड़ जाते हैं, कभी मतलब बहुत इजी क्वेश्चन रहेगा और ब्लैंक हो जाते हैं, क्या बोलना है समझ में नहीं आता है सर. तो
Okay.
एक ये है और थोड़ा
Yeah.
तेज मैं बोलता हूँ ऐसा मेरे को फील हो रहा है.
तनवीर, आपके पहले सवाल के लिए जवाब ये है कि ये सबके साथ में होता है कि जब आप इंटरव्यू में जाते हैं तो आपका माइंड पे बहुत प्रेशर होता है, और एट दैट टाइम ये थोड़ा
यस सर.
मुश्किल हो पाता है कि आप अपने ब्रेन को बहुत ही हाईली फोकस्ड एंड कंसंट्रेट कर पाएँ। आपको बहुत प्रेशर होता है कि
यस
3
सर.
मेंबर्स बैठे हुए हैं, न जाने कौन-सा बम्बार्डमेंट किधर से होगा, आपके पास क्वेश्चंस की बम्बार्डमेंट हो
यस.
रही होती है। और रिकॉलिंग करना
यस.
एट दैट टाइम ये बड़ा टिपिकल टास्क होता है सबके लिए। और जैसा कि अभी आपने बताया कि अभी जस्ट आपने रिसेंटली एमएससी कंप्लीट किया हुआ है, एंड आई थिंक यू
यस
आर
सर.
जस्ट ओनली 23 24 5 4 और 5 ईयर ओल्ड, राइट?
23 सर.
23
23
ओल्ड,
राइट नाउ.
ओके। सो
यस.
ये होना ऑब्वियस है कि आपको थोड़ी सी नर्वसनेस होगी, बट उसके लिए आप क्या कर सकते हैं,
यस.
वो सजेशंस रहेगा कि हो सके तो इंटरव्यू में जाने से पहले आप अपने फ्रेंड्स को, अपने कलीग्स को, या फिर अपने भाई को, किसी को भी ऐसे 2-3 बार आप इंटरव्यू को दे दीजिए, ताकि आपके मन में जो हेजिटेशन
यस
है,
सर.
जो फियर है, वो क्या हो, थोड़ा गायब हो जाए। एंड होपफुली कि अभी आप ये वाला
यस सर.
इंटरव्यू फिनिश करेंगे ना, तो आपकी काफ़ी कुछ इंप्रूवमेंट देखने को मिलेगा, और आपको थोड़ा सा कॉन्फिडेंस
यस
बिल्डअप होगा
सर.
कि हाँ, अभी आप बेहतर कर पा रहे हैं। दूसरा,
यस सर.
जब आप आंसर करते हैं तो ऐसा थोड़ा-सा मुझे लगा कि 2 वर्ड्स के बीच का जो गैप है टाइमिंग का, वो थोड़ी कम है।
यस सर.
शायद जो
यस
इंटरव्यूअर
सर.
होंगे उनको आपकी पूरी बात को समझने में हो सकता है कि प्रॉब्लम
यस.
हो जाए। तो मैं सजेस्ट
यस
आपको
सर.
ये करूँगा कि जब आप आंसर्स को कर रहे हों, थोड़ी हड़बड़ी में ना करें।
यस.
आराम-आराम से ईज़िली, जैसे
यस सर.
मैं अभी आपको बता रहा हूँ, ये समझा रहा हूँ, तो उस हिसाब से अगर आप बोलेंगे तो आपकी
यस सर.
बात भी क्लियर होगी, एज़ वेल एज़ ये
यस सर.
देख के भी लगेगा कि आप कॉन्फिडेंट हैं। ऐसा लग रहा था कि जैसे अब जब आप आंसर्स कर रहे थे, तो जैसे पूरा दिमाग में हड़बड़ी मची हुई है कि जल्दी से बस ये बात को फिनिश कर देना है और खत्म
यस
करके
सर.
निकल जाना है ना?
यस सर. यस
तो
सर.
वो वाला मैसेज,
एक्चुअली दैट टेंशन बिल्डअप हो जाता है, लगता है कि
या एग्ज़ैक्टली
आंसर सही दे
वो
रहे हैं
चीज़
या
वो
गलत दे
चीज़
रहे
भी
हैं,
था
सब
मुझे
सब
कि जब आप
प्रोसेस माइंड
आंसर्स
में.
कर रहे थे तो ऐसा लग रहा था कि हाँ, सारी चीज़ें फटाफट दिमाग में फ्रेम करना है, उसको पूरा बोल देना है, अदरवाइज़ सब भूल जाएँगे और सब बता नहीं पाएँगे। तो वो आपको
यस सर.
थोड़ा-सा मैनेज करना पड़ेगा। इंटरव्यू आपका कब है, 11 को?
सर 10 को 9:30 पे उनका
अच्छा, 10
रिपोर्टिंग
तारीख
टाइम
को
है.
है, ठीक है। तो अभी आपके पास 8 का
यस
और 9
सर.
का टाइमिंग है। कोशिश आप ये कीजिएगा
यस.
तनवीर, आपकी कम्युनिकेशन ठीक है, और मतलब आपकी सब्जेक्ट पे भी पकड़ बहुत अच्छी है। मतलब आपने जो टॉपिक चूज़ किया हुआ, लाइक है इम्यूनोलॉजी, जेनेटिक इंजीनियरिंग, आरडीटी पे तो सवाल ज़्यादा
यस.
मैंने आपसे नहीं पूछे, बिकॉज़ आपने उधर की तरफ थोड़ा वो किया, बट इम्यूनोलॉजी पे आपकी पकड़ ठीक है, अच्छी है। और आप
यस.
लखनऊ के बारे में भी जानते हैं, बिहार के बारे में भी जानते हैं, होपफुली केरला के बारे में भी जानते ही होंगे। आपका ये जो
यस सर.
इंटरव्यू होने जा रहा है ना, देखिए वहाँ पे क्या होगा कि आपसे सिर्फ़ आपकी जो पढ़ाई है, स्टडी है, उसको उससे लेकर सवाल नहीं होगा, ठीक है?
Yes.
मेन जो
Yes.
टारगेट है आपके इंटरव्यू का वो क्या है कि आपका इंटेलेक्चुअल कैसा है, आप पर्सन कैसे हो,
Yes,
और
sir.
आप किसी चीज़ को डील कैसे करते हो, ये फाइंड आउट करना है।
Yes.
नॉलेज का जो
Yes.
चेकअप था वो आपने रिटन एग्जाम में प्रूफ कर दिया दैट यू हैव अ गुड नॉलेज अबाउट द
Yes.
बायोटेक्नोलॉजी, सो देयर इज नो एनी डाउट।
Yes.
So the thing is that
Yeah.
ki, is there any matching of your knowledge, bookish knowledge and your intellectual? So that will be the main prime
Yes.
duty of the interviewer to find out the suitable candidate that has a good intellectual as well as the good concept of the knowledge, राइट?
Yes.
तो ये चीज़ की उनकी ज़रूरत है। सिर्फ पढ़े लिखे लोग, मतलब सिर्फ जो किताबी नॉलेज लेकर के आएंगे उनकी ज़रूरत नहीं है, बिकॉज आप एक ऐसे पोस्ट के
Yes,
लिए
sir.
अपॉइंट किए जाएंगे जहाँ पे आपको पब्लिक इंटरेक्शंस होंगे, डील करना पड़ेगा, बहुत पैनिक सिचुएशंस भी आ
Yes.
सकते हैं, आपको टास्क कंप्लीट करने के लिए बहुत प्रेशर भी हो सकता है, आपको एक टाइम पे मल्टीपल टास्क दिए जा सकते
Yes,
हैं,
sir.
तो इस तरीके का चीज़ होनी चाहिए। हालांकि उसके लिए आपको ट्रेनिंग मिलेगी,
Yes.
ऐसा नहीं है कि आप उसके लिए ट्रेनिंग नहीं दे पाएंगे, लेकिन उनका जो प्राइम ड्यूटी होगा
Yes.
वो यही होगा कि एक ऐसा कोई बंदा मिल जाए जो कि रिटन एग्जाम में भी नंबर पाया हुआ है और उसकी इंटेलेक्चुअल भी अच्छी हो, वो प्रेजेंट दिए गए
Yes,
सिचुएशंस
sir.
को अच्छे से टैकल कर पाता है, उसके खुद के अपने ओपिनियन है, थॉट
Yes.
प्रोसेस है किसी चीज़ को लेकर के कि हाँ, अगर ऐसा है तो क्या होगा, अगर
Yes.
ऐसा है तो क्या होगा, राइट? तो वो
Yes.
चीज़ ठीक है, मतलब आपने ऐसा नहीं कि हाँ, आपने मुझे ऐसा नहीं लगा कि किसी सवाल में आप रुके हैं और मतलब कुछ-कुछ क्वेश्चंस रहे हैं जहाँ पे आपने डिनाई किया है, वो ऑब्वियस सी बात है, होता है, सबके साथ में होता है, तो दिक्कत नहीं है। सब्जेक्ट पे आपको दिक्कत नहीं है तनवीर, सब्जेक्ट पे आपने जो भी बोला अच्छा बोला है, और बाकी
Yes.
थोड़ा 1-2 चीज़ मैं आपको और बताऊँगा जिस पे आपको काम कर लेना चाहिए, लाइक एज़ अगर मैं आपसे पूछूँ कि आपकी सबसे बड़ी ड्रॉबैक क्या है? कमी क्या है आपके अंदर में जो कि आपको लगता है कि इसकी वजह से आप ब बहुत जगह
Yes.
पे आपको नुकसान होता है? तो उन कमी के बारे में नोटिस कर लीजिएगा। कौन से वो पेपर्स हैं या
Yes.
सब्जेक्ट हैं या वो टॉपिक है जिससे कि आपको बहुत डर लगता है? उसको आप नोटिस कर लीजिएगा, कौन-सा वो टॉपिक है, पेपर है या
Yes.
एरिया है जिस जो कि आपको बहुत ही कॉन्फिडेंस है, जिस पे आप बहुत अच्छा फ़ील करते हैं, वो भी आप पता कर लीजिएगा, राइट? इन सारी चीज़ों पे थोड़ा वर्क
Yes.
कर लेंगे तो आपको थोड़ा ठीक रहेगा। दूसरा सपोज़ कि
Yes, sir.
अभी आप प्रोजेक्ट में जुड़े हुए हैं और डेफ़िनिटली अगर आप यहाँ पे
Yes.
सेलेक्ट होते हैं तो ये प्रोजेक्ट आप क्विट कर देंगे, छोड़ देंगे। लेकिन मान लीजिए कि अगर
Yes, sir.
आप सेलेक्ट नहीं होते हैं, देन उसके बाद आपके पास क्या ऑप्शन होगा? इस सवाल का भी जवाब आप आसानी से
Yes.
रख लीजिएगा कि सपोज़ इंटरव्यूअर विल आस्क टू यू दैट व्हाट यू विल डू आफ्टर इफ यू आर नॉट गेटिंग सेलेक्टेड हियर। तो आपको आंसर ये देना होगा
Yes,
कि
sir.
हाँ सर, मैं क्या करूँगा? आप तैयारी करेंगे या पीएचडी
Yeah.
कंटिन्यू करेंगे या जो भी करेंगे? तो ये कुछ-कुछ सवाल हैं,
Yes.
ये रिकॉर्डेड वीडियो आपको मिल जाएगा तनवीर, आप इसको अच्छे से 1 से 2 बार देखिएगा। जब आप देखेंगे ना तो
Yes, sir.
आपको लगेगा कि हाँ, जहाँ पे आपको ज़रूरत है, थोड़ी-सी आपकी स्पीड जो है वो कम करनी है, थोड़ा-सा, ठीक है? आपको आराम
Yes,
से
sir.
पॉज़ लेकर के बोलना है।
Yes.
और दूसरा ये है
Yes.
कि जो जो चीज़ मैंने आपको सजेस्ट किए हैं, इन पे आप थोड़ा-सा ध्यान दे लीजिए, मतलब उन पे थोड़ा एक वर्क कर लीजिए, रफ़ आइडिया कि क्या-क्या आपके डिमेरिट्स हैं,
Yes.
क्या-क्या आपके मेरिट्स हैं, क्यों आपको लगता है कि आप इस
Yeah.
जॉब के लिए एक अच्छे कैंडिडेट हैं, राइट? इस तरीके की जो चीज़ें हैं।
Yes.
बाकी आपका मतलब कम्युनिकेशन ठीक है, सब्जेक्ट पे पकड़ आपकी अच्छी है, कोई दिक्कत नहीं है, और ड्रेस भी आपने बहुत ही अच्छा पहना हुआ है, तो आप ऐसे ही लाइट कपड़े पहन कर के जाइए,
Thank you, sir.
है ना? और बहुत खुशी हुई तनवीर कि आप जस्ट 23 ईयर के हैं और आपके पास 2-2 3-3 गवर्नमेंट जॉब के ऑफ़र हैं, लेटर हैं, रिज़ल्ट वेटिंग में हैं, और आप पूरे ऑल ओवर इंडिया से बायोटेक्नोलॉजी के पोस्ट के लिए सेलेक्ट किए गए हैं इंटरव्यू के लिए। आपको अपने आप में काफ़ी गर्व फ़ील होना चाहिए कि इतनी कम एज में, जहाँ पे लोग आज की डेट के बच्चों को मैं देखता हूँ तो 23 ईयर्स में जस्ट वो बाइक राइडिंग करते हैं, मौज-मस्ती करते हैं और गॉसिप्स करते हैं, और आप इतनी कम उम्र में इतने ज़्यादा फ़ोकस्ड हैं चीज़ों को लेकर के, तो मैं तो पहले इसी चीज़ को एप्रिशिएट करता हूँ कि रियली अगर आप ऐसे
Thank
ही
you,
रहें, ऐसे ही इस तरीके से फ़ोकस अगर आप रहेंगे तो आई मतलब मुझे नहीं लगता कि आपको 25 से 30 साल के अंदर, मतलब 25 साल तक आपको जो झेलना पड़ेगा, कहीं न कहीं आप सेलेक्ट हो जाएँगे डेफ़िनिटली। तो ये आपका जो मैच्योरिटी और अपने करियर को लेकर के जो आपका डेडिकेशन है, ये बहुत ही अच्छा है। इस एज में जहाँ पे लोग एन्जॉय करते हैं, आप पूरी तरीके से फ़ोकस्ड हैं और आपको इंडिया का और आपके फ़ील्ड का, बायोटेक्नोलॉजी का वन ऑफ द ग्रेटेस्ट जॉब की अपॉर्च्युनिटी है, कॉल लेटर आपके पास है, और माशाअल्लाह अगर भगवान ने सब कुछ ठीक-ठाक रखा तो आप सेलेक्ट अगर हो जाएँगे तो आपके लिए बहुत बड़ी बात होगी, ठीक है ना? तो तनवीर बाकी सब
thank you.
ठीक है, बस जिस चीज़ पे मैंने आपको बा बोला है उस पे आप ध्यान दे लीजिए, उस पे वर्क कर लीजिए, और अगर आपको
जी सर।
लगता है कि कोई चीज़ के लिए फर्दर आपको ज़रूरत है तो आपके पास नंबर है, आप उस पे मैसेज कीजिए। डेफिनिटली आई
Yes,
विल
sir.
सपोर्ट एंड
Sir, one more thing I would like to ask one more thing.
या
Sir, actually, when I'm talking in English, certain time I have felt that कभी कभी वर्ड्स हमको ऐसा होता है कि वर्ड्स मिलते नहीं, लाइक क्या बोलना है, वर्ड क्या यूज करना है, मैं भी बहुत बार पॉज हो गया था, मैं लाइक
देखिए ये प्रॉब्लम,
आई एम नॉट गेटिंग द पर्टिकुलर ऐप वर्ड।
या ये
तो
प्रॉब्लम,
इज इट ए नेगेटिव पॉइंट फॉर मी?
नेगेटिव पॉइंट्स आप इसको मत बोलिएगा, बिकॉज ये सबके साथ में होता है। उसके पीछे कारण ये है कि वी ऑल आर द इंडियंस एंड इंडियन डजंट हैव अ मदर टंग लाइक एज इंग्लिश, राइट? सो बेसिकली
यस।
होता ये है कि जब आप किसी चीज़ को सुनते हैं और उसका आंसर्स आप समझते हैं तो पहले उसको फ्रेमिंग करते हैं बनाने बोलने के लिए, और इन द प्रोसेसिंग
यस।
दैट इज जो आपके पास कॉन्सेप्ट है और उसको आपने फ्रेम किया किसी दूसरे लैंग्वेज में बताने के लिए, उस बीच में यहाँ पे लैगिंग हो जाती है। शायद वही जवाब अगर आपको
यस।
हिंदी में देना होगा तो ये प्रॉब्लम आपको नहीं आएगी जो अभी आपने एडमिट किया हुआ है।
या।
सो इट्स नॉट योर
यस सर।
नेगेटिव पॉइंट, इट्स जस्ट अ प्रॉब्लम ऑफ लैंग्वेज। और डेफिनेटली हम सभी लोग प्रोफेशनली चाहे जितनी इंग्लिश बात कर लें, लेकिन नॉर्मली जब हम होते हैं तो हम हिंदी में ही बात करते हैं, राइट? तो वो हिंदी हमारे
यस।
जहन से जाती नहीं है, तो जब हमें कभी ऐसे ओकेजनली प्रोफेशनली इंग्लिश बोलना पड़ता है तो हमें लगता है कि अच्छा हमें सोचने की ज़रूरत है, तो वो होता रहता है, ऐसा कोई प्रॉब्लम नहीं
यस।
है। दूसरा चीज़ ये भी होता है कि कुछ वोकेबलरी टर्म्स जो होते हैं वो उनकी कमी पड़ जाती है कभी कभी। आपके मन में होता है कि हमें ये
यस सर।
बोलना है, लेकिन उसके लिए अप्रोप्रिएट वर्ड नहीं मिलते हैं, तो वो लैकिंग है उसका।
या।
और वो चीज़ तब आएगी जब आप उसके डिफरेंस में कई सारे ऐसे हैबिचुअली अगर आप हो जाते हैं बुक को पढ़ते 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 पढ़ते, तो उससे रिलेटेड जितने टर्म्स होते हैं ना वो धीरे धीरे आपके दिमाग में आ जाते हैं, तो आप उसको यूज़ करना शुरू कर देते हैं।
यस।
और अभी आपकी एज बहुत कम है तनवीर, और इस एज में भी आपने इतना कुछ अचीव किया है, ये बहुत अप्रिसिएबल है। आप और स्टडी
थैंक यू।
करते रहेंगे, स्टडी करते रहेंगे तो डेफिनेटली आपके पास वो वोकाबलरी भी हो जाएगी, जिससे आप अच्छे तरीके से सेंटेंस को बना पाएंगे और अप्रोप्रिएट वर्ड भी यूज़ कर पाएंगे। तो उसको आप नेगेटिव में काउंट मत कीजिए, वो नेगेटिव पॉइंट नहीं है। इट्स जस्ट अ प्रॉब्लम ऑफ लैंग्वेज, नॉट अ नेगेटिव थिंग्स, ओके? और आपको बता दें कि कई सारे लोगों का इंटरव्यू मैंने अभी तक ले लिया। बहुत सारे लोगों का वीडियो अपलोड नहीं करता हूँ क्योंकि उनको थोड़ा दिक्कत होती है। तो इंग्लिश की
यस।
प्रॉब्लम जो है वो इंडियंस में रहती है ना? ऐसा नहीं है कि किसी में नहीं रहता है, थोड़ा किसी में ज़्यादा रहता है, किसी में कम रहता है। और एक्चुअली में इंटरव्यू
यस।
देने से परेशानी नहीं है, एक्चुअली में लोगों को ये इंग्लिश में बात करने में ही प्रॉब्लम है, मेजॉरिटी लोगों को, है ना?
यस सर।
उनको इंटरव्यू का प्रॉब्लम नहीं है। इंटरव्यू अगर हिंदी में हो तो बहुत कंफर्टेबल लेंगे, सर पूछिए क्या पूछना चाहते हैं आप, हम आराम से बताएंगे, जो आएगा तो आएगा। लेकिन चूंकि उनको
यस
उस
सर।
टाइम पे अपने आप को एकदम कंटिन्यूसली इंग्लिश में बात करना होता है, पड़ जाता है, इसलिए थोड़ा सा वो नर्वस हो जाते हैं। तो ऐसा नहीं है, बाकी सारी
या।
चीज़ें ठीक हैं, ओके? सो तनवीर, अगर आपको फर्दर कुछ दिक्कत रहेगा तो आप ज़रूर मैसेज कीजिएगा, और आपको हमारी तरफ से ढेर सारी बेस्ट
यस सर।
विशेस एंड बेस्ट ऑफ लक फॉर योर इंटरव्यू। अच्छा इंटरव्यू देकर के आइए, अच्छा होगा, आपके चीज़ें बहुत अच्छी हैं। देने के बाद आप ज़रूर अपना जो एक्सपीरियंस होगा उसको शेयर कीजिए, ताकि बच्चों को भी, बाकी जो लोग हैं उनको भी,
डेफिनेटली सर, डेफिनेटली।
है ना?
यस।
चलिए, ओके। अच्छा लगा, नाइस टू टॉक
ओके,
ये,
थैंक यू सर।
या, गुड नाइट।
थैंक यू सर, गुड नाइट।